Hello, welcome to Automated Preparation of Nerve Tissue for TEM. My name is Thomas C. Strader. My co-author is Benjamin K. August. I'm from Microscopy Innovations in Madison, Wisconsin, and Ben is from University of Wisconsin School of Medicine and Public Health in Madison, Wisconsin, where I am also an honorary associate fellow. Nerve tissue is imaged using transmission electron microscopy for myriad reasons, including diagnosis, research, and education. A new system was developed by scientists at UW-Madison and Microscopy Innovations that can process from 1 to 24 specimens from fixed rinse through infiltration in 37 minutes, fully unattended and with excellent results, and is also easy to clean up and easy to set up. The ASP1000 is a benchtop specimen and tissue processing robot. It is designed to process tissue specimens or grids. The grids are used for post-staining or immunogold labeling or negative staining. The samples are imaged on TEM or SEM instruments. The processing is fully unattended and the results are very consistent, very robust, very reproducible. Many biological tissue specimens pose a unique challenge because of their small size and lack of rigidity, which is the case with rat nerve. The researchers at UW-Madison and Microscopy Innovations have developed a method to process these small, delicate samples rapidly in a walk-away fashion. Perfusion-fixed rat tibial nerve specimens were collected and stored in half-strength buffer. The specimens were removed from the buffer and placed in specimen capsules from Microscopy Innovations for safe and secure processing. Here you will see on the left a picture of an actual ASP1000 robot with the 6-position deck. On the right, you will see the virtual tray map of the 6-position deck. You will see each tray, 1, 2, 3, 4, 5, 6, is represented 1, 2, 3, 4, 5, 6 over here on the tray map. Here you will see osmium rinse, urinal acetate, ethanol for dehydration, fixed rinse, and resin steps. The barcode label capsules are loaded with the tibial nerve specimens and mounted on the ASP1000. The ASP1000 deck was preloaded with reagent reservoirs containing all of the reagents and rinsing solutions needed to prepare the specimens for imaging by TEM. There are a number of types of reservoirs that can be used on the deck of the ASP1000. For viscous reagents such as resin, we use what's called a 12-column trough. Once the specimens are mounted to the shafts of the ASP1000 and the reagents are pre-dispensed and loaded into the correct positions, the robot, unattended, performs rinse of the fixative buffer, osmification and rinsing of the osmium, urinal acetate staining and rinsing of the urinal acetate, dehydration with ethanol and acetone as a transition solvent, and then infiltration with various increasing amounts of resin. Notice here the fast agitation of the EPON type resin in the video. You will notice it's exchanging the resin approximately once per second. Here we have the timetable of the protocol that we used to process the rat nerve. You will notice that the fixative rinse steps each were a half a minute long, and during that half a minute there were 25 fluid exchanges, approximately 1 every 1.2 seconds. There were 3 fixative rinses in Sorensen's fix, and then it was osmicated in 2% osmium for 3 minutes. During that 3 minutes, there were 150 exchanges of the osmium, which means the osmium was aspirated. 100 microliters was aspirated into the sample chamber, the MPrep-S capsule, and that 100 microliters was dispensed back out into the reservoir where it was held, and then it was aspirated again, and that occurred 150 times over that 3-minute period. And then the osmium rinse occurred for a half a minute, 30 seconds. During that 30 seconds, there were 25 rinses occurred. And then it did a 2% urinal acetate step. The urinal acetate, like the osmium, was in water. It was in urinal acetate for 2.5 minutes, which was 125 fluid exchanges in the 2.5 minutes. And then there were 3 rinses, 1/2 minute each of water after the urinal acetate step. And then we proceeded on to the ethanol dehydration steps. One thing I should note, we have lately moved to acetone dehydration and gone straight from there to resin and had very good luck. In this case, we did ethanol dehydration, used acetone as a transition solvent, and then went into the resin. We never use propylene oxide. There's a safety hazard with it. We may have to do some modifications to your protocol if you are using it now and you decide to move to an ASP 1000 platform. We've done that quite often. It has not been a problem. Back to the protocol, we do the acetone transition step, and then we move into the resin step. The resin steps are about 3 minutes each, with the 100% resin steps to 4 to 5 minutes. And then there's an overnight resin step in the oven at 60, which is polymerization. You'll notice at the bottom the total time is 37 minutes. Over that 37 minutes, there were 1,850 fluid exchanges of various reagents and rinsing solutions and dehydrants and resins. Following the 37-minute robotic processing protocol, the capsules were polymerized overnight at 60 degrees. The next morning, the capsules were trimmed and sectioned. No post-staining was performed. So here we have 4 images collected in a Philips 120 stem at 4 different magnifications: 1 micron, 2 micron, 8 micron, and 10 micron reference bars shown of tibial nerve bundle. In the bottom right panel, you can see the white axon in the middle, surrounded by well-defined dark myelin sheath, with Schwann cells enveloping the lower half of the cross-section. We're running short on time, so here's another beautiful nerve image. You'll notice again the beautiful myelin sheath with the nerve in the middle. And one more beautiful image. Okay, well, that's going to wrap it up. Thank you very much for attending my talk. Appreciate it. If you have any questions, please contact me at my number on the slide there, 608-770-7649, or send me an email, tom.strader@microscopyinnovations.com. Thank you, and have a great day.